Hello and welcome to the very first episode of Double Stranded Podcast, a podcast where we'll talk about everything related to genetics. In today's episode, I sit down with Dr. Raquel Cuella Martin, whose work is mainly focused on genome editing and DNA damage response. She's a brilliant scientist, and I cannot wait to share our conversation with you. Thank you so much for coming. It's so good to see you. I wanted to first congratulate you on your official lab opening, which is such a huge accomplishment. I actually read somewhere that only 1 in 10 grad students will actually make it to run their own lab one day, which is really huge. So how's the recruitment process going for your lab?
I think that's going well. I am quite happy with the students, my current student and the upcoming fall 2023 cohort. Recruitment in other areas is much more tricky. We have a shortage of postdocs.
Exactly.
So that's an ongoing search. But, we are doing some science, as you were saying. We are now up and running. So it's exciting to see that.
For sure. So you yourself did your postdoc at Columbia University, right?
Yes.
And so it says here that you used CRISPR-dependent base editing to perform genetic screens, right?
Yep.
So let's break this down. What is CRISPR? What is CRISPR-dependent base editing? What is base editing? And what is genetic screen?
Okay, so, I'll start from the first word.
Yeah, CRISPR.
I think that by now, since this CRISPR work came to our lives as this tool for engineering our genome, with that flagship publication 2012, 2013, and I guess received even more buzz with the Nobel Prize that was awarded to Doudna and Charpentier. I think everybody, many people that are not in the field, and I talk to them, they're like, oh, that's the thing, those scissors, or the genetic scissors. And I'm like, exactly, that's what it is. It's a very powerful tool to do genome engineering. But if I follow on that CRISPR-dependent base editing, the CRISPR Cas9 system, that was the one that was traditionally used, is more technicalities in here, is an RNA-guided endonuclease. So it will go to our genome and cut. And that way we can disrupt the gene function. But the base editing is a new engineered protein, a new engineered version of that Cas9 that goes to our genome and has another enzyme coupled to it and makes a mutation. So instead of now having a cut, we are making a single point mutation in a specific region. And I guess that was really cool. That development came later, 2016, 2017. And very interestingly, because, the majority of our genetic variation is just single point mutations. It's not full of loss of a gene. It's just single changes. So that's the power of this technology. That's what base editing means. Base, obviously, we change a base at a time, a DNA base at a time.
So before this technology, we couldn't do editing down to one base?
So we could. There was another technology that used the Cas9 as well, made a cut, and then you could put a donor template carrying that specific mutation and trust that the cell will pick that template that was exogenous, think that it's real, homologous, sister chromatid, and insert that mutation in the genome. We could do that. It's still in use, of course, but low efficiency and highly dependent on the cell repair mechanisms, and also still we generate a cut on a gene that it can either result in a knockout as well. So, but we had that technology already in place. But the level of efficiency that base editing and versatility of using different cellular models that base editing gives was not there before. So I think it was a clear step forward.
What about this genetic screen? Because I guess you do all of that to perform genetic screen, right?
So I think what we do with base editing is what we call genetic screens at nucleotide resolution.
Yeah.
So when you think about traditional Cas9 CRISPR knockout screens, our resolution is like gene level. So you knock out a gene, you inquire the function. But we work with genes that are complex, that have multiple functions, that are large, that have, you have a function that is driven by one part of the protein, another function driven by other part of the protein, multiple interactors. So with this base editing, instead of gene level, is nucleotide level. We do genetic interactions between mutations. So you go, instead of full knockout, you mutate the gene from beginning to end. And you inquire phenotype-genotype associations when the genotype is not a knockout, is a mutation. So that's obviously moves the level of resolution to whole gene to specific amino acids. And that's that's what it's our move forward. And what we call with cell genetic screens is just like, because we do genetic modifications at high throughput level, and we screen for a specific phenotype, so to be able to draw those genotype-phenotype associations.
Is this kind of like similar to what you're doing right now in your lab?
It is, or at least what we want to do. We have started, yes, and we see a lot of power on the technology. We see a lot of genes that are not very thoroughly characterized because they are tricky to work with, and we see that technology can give us a lot. And actually, when you inquire, you make the mutations in situ, what we call in the endogenous loci, you can inquire non-coding elements, you can inquire splice sites, you can alternative splicing. That it's something that is a whole new thing that we can do now. Or the gene regulatory elements is also a whole new thing that we can do now. So yeah.
So I was doing a little bit of research, and apparently there is something called prime editing as well, and like base editing. I guess we kind of talked about this one. So what is this prime editing?
I was telling you before about base editing. You have a version of the Cas9 fused to a second protein that makes a point mutation. And in the case of base editing, it's a cytidine deaminase or an adenosine deaminase. In the case of prime editing, the second protein that you have is a retrotranscriptase. And why a retrotranscriptase, right? The Cas9 is guided to the specific sites of the genome where we want it to edit with an RNA. So it's an RNA-guided endonuclease. So one will think, like, and very smartly, the David Liu's lab thought, what if we extend that RNA, include a template that the retrotranscriptase will recognize and think, okay, I can synthesize DNA that is homologous to that template. So when you take the whole system to a specific site of the genome, that retrotranscriptase starts synthesizing inside our genome based on the template that we are inserting. So instead of only single nucleotide variants, we can now insert small insertions. We can do small deletions. So, that's a bit expanding a bit more our toolbox for specific edits.
Is this based on CRISPR?
It's based on CRISPR, yeah. Okay. You have a Cas9, a nickase version of the Cas9 fused to that retrotranscriptase. And the Cas9 acts as like the guide for that retrotranscriptase to the specific sites.
So these are all like different versions of CRISPR?
Yeah.
Okay, great.
Yeah.
So like before CRISPR, what was the history of the field? Like what were people doing before CRISPR?
I think immediately before, or the thing that was a bit more similar, were like there were other types of nucleases. People were using like zinc finger nucleases or the TALEN systems to make to do a bit what CRISPR is doing to make cuts in the genome. There was also a lot of recombination-based systems. For instance, the way the transgenic mice were generated, that was all recombination-based. it was not like we couldn't do genome editing before.
Yeah.
But we went from like, okay, you had to have a lot of expertise. You had to have like, it was more laborious. It was like pretty challenging, and to have something that we could do in any lab at any time with much more ease and much more efficiency. So that's,
So now that we have CRISPR and all these technologies, what are the main applications?
I take your question more towards clinical applications. There are so many things that it's hard to pick one. No, so we can do a lot of things on the drug development area. People have worked we have used CRISPR and CRISPR screenings for target identification. So like what are specific tumor vulnerabilities using inquiring the effects of knockouts in tumor cells. But I think for me, one of the most exciting applications comes from in the field of gene therapy. Since CRISPR started, people saw the value on that, and now even with base editing and prime editing, I think even more, they are very efficient genome editors. So if you suffer from a genetic condition that is caused by a specific mutation, that might be able to be corrected. And that is where this kind of gene therapy is going forward. Like, can we do that? Can we actually engineer those CRISPR systems and deliver them in an efficient manner in an adult and be able to correct a genetic defect? And
yes, so you mentioned drug development. Can you give us some examples of the projects that used CRISPR in drug development?
Okay. I will say that one of the things that, where I think CRISPR has been heavily used is in what we call the synthetic lethality field. So if you have a tumor, and I will talk about cancer research because that's a bit more I always lean towards that. If you have a tumor and the tumor has a specific gene amplification or has specific gene loss, okay, how can we exploit that specific gene loss that is not present in normal cells? And with that, you put a whole genome library, you knock out every single gene of the genome individually, and you say okay, which combination of that genomic alteration or epigenetic alteration that the tumor is carrying with a genetic knockout is giving us a synthetic lethality phenotype, meaning now the cells are dying much more than with the individual perturbations. And, if you find that instance, now you have a drug, you have a target. And that has been used. And then there are companies, even here in Montreal, that are developing drugs based on targets identified using these CRISPR screenings. And that moves us towards more what we call the personalized medicine. And you have this tumor with this specific genetic alteration or epigenetic alteration, we can give you this drug because those are vulnerabilities that are being directly targeted.
But the way that you described it, it sounded so expensive and labor intensive to check every possible. Is it like this?
I guess with CRISPR it has become much less labor intensive to do something like that. I will say that for what you get out of it is not labor intensive at all. But yes, when you have to sit in tissue culture for many hours to do those screens, they are not, they are laborious. They are big experiments. They tend to be long. But no, the way they are conducted is you do it in a group of cells in pooled populations. And we have strategies to then inquire the effects of the individual phenotypes or tease them apart by using next generation sequencing. I think it has significantly simplified our way of doing screenings. And it's not like we were not doing them before. We have been using siRNAs, shRNAs for inquiring whole genomes before, but now we can do genetic knockouts, and that gives us a next level of confidence.
How much of the whole process can you automate?
I think bottom line here is that the tissue culture part, that's not
Or
very
like, is
easy,
it something
very
that
easy to automate. That's what takes us more. That's where we sit in the hood for a long time. Readout measurement, that can be automatized. Like, of course, if you have library preparation and you can do fluidic, use fluid.
But is it like something that people care about? Like, I don't know, a group of researchers like working specifically to like simplify and like the whole process?
I mean, yeah, we tend to always try to simplify the processes, and yes, there are. Yeah, I would say so that everybody that works a bit more on technology development and a bit more on the process development area, they tend to work from what is the best Cas9 variant or the base editor that you can use that gives you the best definition of, like, the best resolution for your phenotypes, to what are the best timelines, when can you get the same quality of data with like shorter screens versus longer screens. We do that. We do that because we want to have everything as optimized as possible. That's always what we lean towards.
So how do you go about doing gene therapy with this technology?
So if you think, I'll talk about base editing because that's a bit more where I am at. Base editing the papers were 2016, 2017, and people started thinking on how to use it for gene therapy. We now have gene therapy in clinical trials using base editing to actually a gene probably that you have heard of many times, to correct PCSK9 for hypercholesterolemia, familial hypercholesterolemia, and that went already through the preclinical, the mouse models. There was work done in in non-human primates, and then that moved to clinic in the span of we'll say 5 years, 6 years tops. How do you go about it? Well, the current technology is it's ribonucleoprotein based delivery. So you deliver the base editor, you deliver the guide RNA that will lead that base editor to your specific region, and then that is systemically delivered to the person. In the work that was done in monkeys, they observed that they got reversion of the genetic alteration in the liver at high rate, and that resulted in a reduction in the cholesterol that was stable for 2 years. So that's I think that's how it has been traditionally, and I think that there are perhaps two clinical trials at the moment that just started moving those therapies forward, and I think it's pretty impressive for the short life of those technologies, like 5-6 years.
Yeah. So you mentioned also cancer therapy, which I guess is one of the more exciting areas that this technology is being applied to. And is this your research topic, cancer?
I work a bit more on cancer, yes.
So maybe we can elaborate on this cancer therapy.
Yeah, I think it links a bit more in cancer. It links a bit more, or what I know, links a bit more with the concept of synthetic lethality that I was talking to you about before. My research is more on molecular mechanisms of anti-tumorigenesis. So not much on identifying that drug targets, but it's equally important. But also, I think, specifically in the base editing realm, we do have quite a lot to say with these technologies for patient stratification, for target drug interactions. You can imagine that if you design a drug you will want to know which patients are going to respond or not. Specifically, if you are targeting a genetic vulnerability, okay, these patients have variants of uncertain significance on the gene that triggers that genetic vulnerability. Are they eligible for therapy or not? And we can do those phenotype-genotype associations with this technology. Or you design a drug and you think it binds this region specifically, but are there other regions in the target that could be influencing that binding? And you can do that characterization also with these technologies. If there is any allosteric sites that could be regulating and directly that binding. And yeah, that's there are a lot of steps in that drug development process, particularly for cancer, that we can tackle a bit better with
this
these
is
technologies.
but the current state of this technology, right?
Yeah.
So what breakthroughs can we expect to see in the coming years? What are you excited about?
I am very excited to see what comes up on all the gene therapy realm, because we are at the stage where you can go to papers and see that more work has been done preclinically, at least in mouse models. There was a mouse model of progeria where the mutation was corrected and the lifespan of that mice was doubled, almost to a normal lifespan. There has been just very recently published very similar work on mice with SMA and how the correction of the mutation largely reverted the phenotype. So when those things reach the point where more preclinical work is done, probably in primates, and that it reaches the clinic, I am like, I really want to see the results of those clinical trials. I really want to see whether we are able actually to effectively cure those patients with a single dose of a treatment in this case. So that I am like, even if I am incredibly excited about my own work, that, and maybe I will move towards that in my research as well, seeing whether we can extend that technology to more and more genetic disorders, and seeing that into clinic. I was like, yeah, that's. I keep an eye on those trials and see what are the releases of data, because I really want to see it.
How longer until this happens?
I think the first trial, the one that I was telling you, for PSSK9, the first wave of results is expected in the 2024. They started recruiting last year, if I'm not wrong, but like phase one, I'm guessing.
Yeah.
So, and seeing other things moving to their clinics, I think in the next year or 2 years, we will have a few more examples already in clinical trials. Now, dosing patients, like FDA approve dosing patients, we might need to wait for a bit longer.
Yeah.
But, towards the end of this decade, we might be seeing a lot of things moving forward.
Yeah, and has any of these things made it to like a clinical trial? Like actually we can like test it on a patient or something?
Yeah. The PCSK9 is on clinical trials. That base editing is already on clinical trials. So yeah, that's why I'm like, I keep an eye on those trials to see whether when they have some results.
Like even in Canada or like?
Oh, it's in the States, that trial. Okay. That trial that I'm talking about, it's on the States. But yeah, we'll see.
Okay, so this is all good, but what are some of the limitations of this technology that we have?
Well,
like technical issues.
I was like, okay, technical limitations. So in the case of CRISPR-Cas9 based approaches, because there are also CRISPR-Cas9 approaches out there for clinical applications, we have the problem of off-targets. So you might be knocking out other genes that you were not intending to, right? Like your RNA molecule that is guiding your Cas9 to your site of interest might bind other regions, in the in how big our genome is, and it's only a 20 nucleotide sequence, it might have other places where it can anneal and take your Cas9 there, and your Cas9 will also cut there. So off-target assessment, I think it's something that is very important, and should be clearly incorporated in what you do when you developed one of those gene therapies. Like there should be a very thorough framework on assessing that before that reaches the clinic. And I think that's a limitation. Another limitation that I will say, we will think about that maybe down the line, is that the Cas9 generates immunogenicity. So obviously it's an exogenous protein. And
what is that?
That, you deliver the Cas9 protein into your system to be able to make that edit. Your immune system is recognizing that as an exogenous protein. So you can generate antibodies against it and mount an immune response against the Cas9. So why this might be a problem? So imagine that you have a genetic alteration and you are eligible for one of those gene therapies. Let's move ourselves like 10 years in the future, right? And then you do it once, your genetic alteration is corrected, but in 5 years, our cells renew, you start seeing that you have symptoms again. Maybe because, you have cells that were mutated, or like some cells that were mutated, and then they kept on repopulating the certain organ or a certain tissue. And then you need a second dose. Okay, if your cells mounted an immune response against the Cas9 the first time, you might not, that second dose might generate an immune response, and actually that Cas9 might never reach the site, and from that second dose you might not get anything at all. And that has been observed in monkeys, that a second dosing was not improving the outcome. They were thinking probably because they mounted an immune response against that exogenous Cas9, and the second time you inject, it's like the immune system is like, this is exogenous, get rid of it. So that's something that, as we have not gone with those trials that long to know if the patient will stay with the mutation corrected and whatever forever, we don't know. But that's
how serious,
that's something that has been
are these
observed.
problems? To the point that it stops the whole research.
I will say the off targets are something that people take a lot into consideration. Like that's, but that's first line when you are designing the therapy. That's one of the first things that you are like, you want to minimize as much as possible. And there are different versions actually of the Cas9 that have higher fidelity. So you need to have a perfect match of your guide RNA with your DNA for that Cas9 to cut. And the moment it binds somewhere else with a mismatch, that Cas9 is not cutting. So those high fidelity variants are being used for gene therapy because it decreases your off target. So there is work and a lot of ongoing work towards minimizing that problem, because that might be something that will tell you like you are not moving forward with this. The way this therapy is built is not safe for the patients. Regarding the immunogenicity, I think that's something that might be, that they may work to avoid, but we still don't know the consequences. And actually, one of the things about base editing or the new technologies is that they minimize, for instance, double-strand break generation. Like the Cas9 doesn't cut anymore, so the cell doesn't respond to that as damage to their DNA. So that minimizes certain problems that the Cas9 has. So I think that as long as the technology development research moves, we get better and better options to translate to therapy as well. And the off targets is something that the base editors are a bit better at, although they have other problems. And just you cannot know. You just need to keep working on these problematics and try to to get it to the best possible point, but at the same time you need to find a compromise between trying to push something to clinic and knowing that it's obviously not in the optimal desirable form that you will have loved for it to be, or with no risk. There might be always a minimal risk of targets, a minimal risk of lower efficiency, a minimal risk of a therapy not working for you. So that's I think that's again as any other therapy that is now in the market.
Yeah. But so like how much of it is like just chance, you can't do anything about it, and like how much of it you can prevent with like better, I don't know, experiment design?
No, I mean, I think that the things that are intrinsic to the technology, that's, if you want to do a Cas9 based approach, that the Cas9 cuts. So that's intrinsic to how the technology works. That's it. You can work to use other advantages or more advantageous developments like base editing or prime editing and don't cut. But that's, if you choose that to move forward, that that's a problem that is intrinsic to the technology, because that's how it works. And then, of course, if you're like, okay, I'm doing a Cas9 based therapy for sure, then you're like, okay, within the realm of how can you design that experiment, let's minimize the other problems. Like off targets are an intrinsic problem, but you can have more or less. So you would go for the less. On target might be an intrinsic problem, but you could work to try to get the optimal on target with like different experimental design. But there are things that are like pure intrinsic to the technology that you are not gonna be able to change much unless you change to another technology or an improvement of that technology. And I think that that's what people are trying to do. Like that, all that work, there is substantial work and like really like by groups that are like out of this world, like extraordinary, on moving the technologies forward and working out those improvements that reduce those technology intrinsic problems that are the ones that we can not avoid really.
Before we move on, I wanted to ask you about this, that how much of genome editing is hype and like how much of it is real? Because I feel like it's kind of like AI, that like the impact is huge and it's real, but also there's a lot of hype around it. So how do you feel about that?
That's a very good question. I think I'm very critical with the hype myself. Like sometimes you do something gets, you have a new discovery, you apply it in the lab in cells, and like the way that gets out to the media is like, this is gonna cure everything. Like, and you're like, no, like we don't know how that can translate to animal models, how that will translate to therapy, how that will, like, we don't know. So sometimes it's like the hype gets generated on results that are promising but small. So I tend to think that you need to be aware of the potential of the technology, but be very realistic about where we are at. The hype is often not generated by the scientists, which that's why we need to get a bit better at communicating and at outreach. The hype is often generated by media. We discover something, I say like, this is very, you write a sentence like, okay, this can be promising for this, that, and that, and immediately the translation is like, this is already done. And you're like, no, that might be, base editing was discovered in 2016, 2017, or was engineered like the first versions of it. Now, 2022, into 2023, we are seeing clinical trials. Of course, it was promising back in the day, but we are seeing clinical trials in one disorder. In like until that, and that is starting the clinical trials. Whether that will work or not, we still don't know. But I think sometimes the hype is justified because the technology is moving fast, and it has a lot of potential. But very realistic about where we are at, and where we are at is, I think, moving very fast, but still being very cautious. until we see that it works. Like the scientists are usually much more cautious until we see that it works.
What about like funding agencies? Are they like more inclined to fund gene editing projects or something?
Tell that to CIHR. I will be very happy if they are inclined to fund my projects. I will say that perhaps yes. I don't know that much about funding agencies because I am, you said it, I'm new to this PI world. So it's my first time going around asking for money. But I will say that, advances in the genome editing field are well received by, for instance, publishing sources, like that they tend to be, really receptive to advances in those technologies. I have found that the Canadian community does not have as much research on genome editing as I would have liked to. And so we will work on that, on moving that forward as much as we can. The more we work, the more we advance, the more things we can put out there, the more receptive CIHR and CERC will be to projects like that, because I do think the scientists are like people are like very keen on collaborating. These technologies are so cool. Like, can we use them? Can we do this and that? But it's sometimes hard to break through the projects. Imagine like if you write a project about developing gene therapy for X or Y, like those projects are high risk.
Yeah.
Or about developing a new tool, even if you come with the expertise, they are like, ooh. So funding agencies sometimes are a bit conservative on that end, but we will fight. We'll fight.
You said scientists are more cautious when it comes to like promising things that are supposed to come with these technologies. But like, what inspired you to pursue a career in genome editing?
Well, I have to say that was a bit serendipity for me. My background and my work, and still my biological interests, are on studying the response to DNA damage. And I was doing that and applying already CRISPR. Like, I started my PhD in 2014, and CRISPR had been discovered just a little bit before, and or had not discovered but had been applied to engineer the human genome. A little bit before, and we just jumped onto that. I made my first experiments on my PhD were making knockout cell lines using that technology, and then we're making like knocking mice using that technology. But that was not my, I was a core pure molecular biologist. And then I ended up on my postdoc, and I had this, maybe we can scale base editing that discovered in 2016, 2017, my postdoc in 2018, starting, maybe we can apply this in a high throughput level, like to inquire our genes, that in the DNA damage response, many genes are tricky to inquire with other approaches. And I was like, okay. And then I ended up working on it, trying to optimize the technology, and on the way there, saying like, this has a lot of potential. This is going to have a lot of applications. We can learn so much about biology. We can learn, we can apply it for this. We can apply it to that. And that's, I wouldn't say that my career is on genome editing, but I would say like, when they're like, you're an expert, I'm like, no. Yeah. But we do use it a lot, and we can see the value of those technologies to move research further. And that's, that's...
What was supposed to be your thesis for PhD initially?
No, my thesis for PhD was always going to be that, and that was what it was. I worked on... the interaction between 53BP1, that is a protein that is involved in the repair of double-strand breaks, and p53 tumor suppression. And that was for my PhD, that was going to be my project. But actually, I think maybe your question was towards my postdoc project. So I moved to the lab in the postdoc thinking of working on like more replication stress, and because it was a replication lab, I will say. But then, they have been starting playing around with those genome editing technologies, branched out, and I ended up not doing any replication work and doing just screenings, and screen optimization and validation and trying to see what we could find with that new technology.
This is a bit random, but can we also edit epigenetic?
We can actually. As part of what I call sometimes the genome editing toolbox or the CRISPR toolbox, people have engineered the Cas nucleases, adding all sorts of things. So, I told you, prime editing has a retrotranscriptase attached to it. Base editing has a cytidine or an adenosine deaminase. So there are other systems that either act on like transcriptional repression or transcriptional activation. You can fuse your Cas9 to a chromatin modifier, and your Cas9 directed by the RNA will go to the places of the genome that you want it to go. And you can go there and do histone deacetylation. You can go that's how you can use the system to try to edit the epigenome as well.
But which one is more important, like editing genetics or epigenetic?
I guess that's depending on who you ask. I work on genetics, so I will say I don't know if I dare to say genetics. It's better not to limit to one or the other. But it's good to see that we have the versatility of being able to do both.
Okay, moving on. What are some of the biggest challenges facing researchers who work in this area? I guess the main one would be like ethical considerations.
I think if you work more on the gene therapy realm, yes, you will have quite a bit to think about on that. I think I like to think that people that work on the genome editing feel like quite in agreement about the ethical considerations. When, I don't know if you have heard about, you probably heard about, because that was a big thing, when scientists in China engineered the genome of human embryos and, those CRISPR babies were born, there was an unanimous response from the CRISPR, the genome engineering community. Like, what are you doing? We don't have the information. We are not at the stage of being able to do anything like that. Technically, we can, but that doesn't mean that we know how that's going to impact, in the long term. And also the consideration of editing human embryos. It's something that it's forbidden in many countries in the world. And you're like, and then the addition there that was for a receptor that prevents HIV entry, like that will, you will, even if you get infected with HIV, you will never develop AIDS. And you will be like, well, that's what was the priority of making that addition. You are not even, you're like, if I ever think to where like, okay, if we reach the point of even considering editing human embryos, the first thing that I will think will be like correcting genetic mutations that are associated with super strong disorders that like the kids have very low lifespans, but not that kind of like I was like, but it was what it was encouraging to see was that the unanimous response from the field. The field was like, what is this? Like, this is just so wrong at so many levels. Like gene therapy is not even there on the clinic yet to edit somatic alterations, and you are like going to the end. Like, no, but the babies are born and they are fine. I'm like, yeah, that's why you did it without nobody knowing, like, because it was so legal. Like, and it's it was quite interesting. I mean, that's it was a good example for us to give us a reality check. What we can technically do doesn't make it correct. Doesn't mean that we have like the right to play with these tools as if they're like no control, with no ethics, with no no considerations. So yeah.
So do you think it's dangerous that like these technologies are becoming more available and like perhaps cheaper? So like many people, like even outside of the field, can like play with these tools.
I don't think that's a bad thing. You know, having a tool like CRISPR being accessible for any researcher, that for me, that was like, that's a really good thing.
Yeah.
The problem is on the researcher end. Like the problem is when the researcher is thinking on doing things that he shouldn't be thinking on doing. Like that's, you can, sometimes we have super powerful technologies. You can use them for things that are really good, and you can use the same technology for something that is really bad. You can like build an atomic bomb, but like the same way that you can like, that's, and it's the same technology, and it's on the morals of the researcher to say like, we have this accessibility, but the accessibility per se, that I can only think that is a good thing. So yeah.
So how do you, as a scientist, balance the desire to make new discoveries and like playing around with these tools with like, as you said, ethical considerations?
Well, I think in the research that I am in, that I do quite a lot more on understanding gene function, I am a bit farther away of the ethical considerations. But when we move to, working on genetic disorders, working with patient cells, working with patient samples, or thinking, you think that it's not all about your research, that there are people behind that have donated the samples, that there are families behind those kids, or like we work a lot with autosomal recessive disorders or the DNA damage response. When you have alterations related to autosomal recessive disorders, and you're like, those individuals that have given the samples, like they have families, they have like they are trying to find a cure or find a how to relieve the symptoms or how to understand better what is going on. So that's not at all costs. Like you are aware that is not okay. I just need this because I want to publish in Nature. No, this is something that we do the research, and if we are aware of our world, we do the research for those people more than even for anybody else. So when we just like stay in our little bubble and it's like, okay, this is only a cell line. I was like, you don't think that came from a person that is suffering from this disease that you're searching. Like, this is not about a Nature paper. This is about making an impact for those people. So yeah, I mean, I think that's, and of course, it's not always, not everything works. Like if that patient consented for X, Y, Z things, I was like, oh yeah, but we already have the samples. Why don't we do it? No. That you have the samples because a person was willing enough to do that for your science and for the scientific community and for the community that is suffering from that disorder, for you to be like, no. I mean, we sometimes, that sometimes people think like the ethical considerations are very strict, but it's better safe than sorry.
Yeah. So in Canada, is it like these regulations are like more or like less compared to other countries?
Yeah, on ethical considerations, I think that's traditionally Europe has always been very tight, and traditionally America has always been a bit looser, or the States, maybe not Canada. So, but yeah, I'll pass the ball to my colleagues, because when you work, I think when you work with patient data or with patient samples more often, then you are more familiar with that I am myself. That sometimes we are like in our little bubble of cell lines in the lab.
Yeah. Some follow-up questions. You mentioned a few times DNA damage response. So like how does this whole thing come into play, like when you think about the whole field of genome editing?
So I think people that were working on the response to DNA damage felt themselves very drawn to this genome editing, and vice versa. Because we are talking about a nuclease is making a break in our genome. That's like DNA damage.
Yeah.
So a lot of things that you can do to make, editing efficiency in the case of genome engineering, make that to make it more efficient is playing with how the cell deals with that damage. To be able to make, I don't know, C2D transitions, you need to play how the cells repair that mismatch that you are generating. And it came all somehow together. Like you needed to know about how those cells will repair the damage that you are inflicting with the Cas9 and with the genome editing techniques to be able to engineer them to go the way you want it. And also, of course, people in the DNA damage response were like, oh, okay, now we have a tool that can allow us to make a single double-strand break and study what's going on in there. Now we have super powerful tools that allow us to study our genes that are, I don't know, 3,000 amino acids or 2,000 amino acids that before were very tricky to study. I think there was like a nice intersection there, and I think people that work on DNA, on genome editing, or started from a pure genome editing background, were like, okay, we need to catch up with the DNA damage. There was like how the cell responds to respond to DNA damage, because this is where we are going to find our tools to make this better.
So what animal models do you use in your research?
I have only used mice and not that much. I shy away a bit from animal models. I don't think I can do that for much longer. But yeah, I during my PhD, I had a little bit of my project that had mouse models, and actually, that's I started my PhD. We made some cell lines, some knockouts, some knock-ins, and then in the span of like 3 years and a bit, we went from not knowing much about what our protein was doing with the other protein to having a mouse model with like a knock-in mouse model already that we could do experiments on. And that's, that's when you realize that these new technologies are making the times from like generating a transgenic mice much shorter, or like generating that mice that carries that specific point mutation like within a PhD that and fairly short PhD. So it was like, okay, wow, right, we discovered this mutation within a year and a half into my PhD. Within 3 years, I'm doing experiments with mouse models. But yeah, only mice for now, and I think that will stay like that.
Some more follow-up questions. How is genome editing being used to study the function of non-coding regions of the genome?
That was one of the things that I get asked, like, how are you going to do non-coding editing? And I'm like, yeah, why not? Like, that's those regions, non-coding elements have traditionally been a bit more tricky because they have repetitive regions that, if you design a guide RNA that will direct your Cas9 in a repetitive region, is probably that is binding here and is binding there and is binding there and is binding everywhere. So it's getting a specificity might be a bit harder. But I mean, there was a really nice paper published, I think, 2021, on using base editing to target the responsive elements that regulate fetal hemoglobin expression, thinking of how to approach sickle cell disease treatment using this technology. And it was a really nice paper. I think it was out in Nature Genetics, and it was really nice to see how you can integrate ChIP-seq, ataxic data, identify those potential elements that regulate the fetal hemoglobin expression, and then go mutagenize them and say, like, okay, where are the sites or the specific nucleotides in those elements, if any, that do regulate the gene expression. And with that, we have enhancers, we have promoters. Sometimes you have a set of elements that you think regulate your gene. You don't know which exactly does or what or in which context. So that's something that we can do. And sometimes, you can change. I was thinking, like, when you think even about how your protein binds more tightly or less tightly, or you have a transcription factor and it has a binding site, but that binding site might have positions where you have multiple options, like fixed positions and then positions that wobble. And you're like, okay, changing those positions, does it make it bind better or worse? Like, can you modulate the activity of that transcription factor just by mutating specific positions within the responsive element? Can we make an optimal promoter for expression? Like those things like that are, are something that we can do moving forward. And I did some work on targeting splice sites, like,
what is that?
Inquire if you target the sites between dextron-intron junctions, whether that generates a full loss of function, because that's sometimes the case. You alter splicing, then the protein is not there, or whether your cell gets around by generating alternative isoforms, or like, okay, well, that would be interesting, very interesting to see. But yeah, that's it's a bit on the non-coding end, and I think it has a lot of potential there, I think.
But I guess that should be really important, because most of the genome is non-coding, I would say.
Yeah, most of the genome is...
So most of the research is actually being done on the coding part?
Of course, yes.
Okay.
Maybe because the non-coding, we know it less. And it's also, I'm telling you, it's a bit more challenging unless you go to, you have your screens or your downstream genome editing experiments guided by, ATAC-seq or ChIP-seq, that you're like, okay, within these regions are regulatory elements. You could go and mutate promoters, like all the 3' UTR, all the 5' UTR inside the introns, and end up with phenotypes all over the place. That's tricky. And also when you think of regions like of heterochromatin, like you target heterochromatin, usually non-transcribed regions, very compact, your chances of getting high editing efficiency are much lower, because obviously you have a accessibility limitation there, and to know whether that will impact or not. So there, I think there is a lot to do on the non-coding genome, a lot. Whether that will give us more information than the coding genome, I don't know about it.
Interesting. How do you see the relationship between basic research and applied research in the field of genome editing?
I believe that now people are thinking much more on the translational value of the things they do, because they see therapies moving very fast to clinical trials. They see that the technology holds a lot of value, both in public and private, like we'll say public and private research, that you do something in the lab and you can identify a target that is druggable. So, and you see seeking much more to move forward that to make a real impact on, like, developing a treatment or a diagnostic test. Or so I think in that field, in other fields, I would say like maybe they are more distant and less connected. In the genome editing field, I think there is a strong push on, okay, get your technology out there, get your target out there, get your things moving forward. Which is really good, but also that's there are big patent wars and things like that in the field, So
do you think we need more basic research right now, or like we have the technology, we should we just need to start applying it in all the possible realms?
I think both. Like we were discussing before that there are caveats and there are things that can always be improved, and a lot of research is being done on technology development in the CRISPR field. Those big breakthroughs like the prime editing, the base editing, all those tools, those are primary, technology development is not, can we apply this? No, it's like, how can we make this technology better? People are working on discovering new CRISPR systems that will increase the versatility of it. And for that, you need to go back to the bacteria or go back to the archaea and be like, okay, we have this one, and this one, and this one. And, maybe other strains will have a different modification. So that research that is still ongoing and that is still moving very fast, it is really important. But I think we have a good balance. A lot of people working on it. I said the field is big, and but yeah, I like to think there's a good balance.
Okay, let's shift gears. What is the most ridiculous conspiracy theory that you have heard?
Oh my goodness gracious! On what? I don't know, but on genome editing.
I mean, I can give you some.
Okay,
Uh,
I want to hear them.
okay. Well, obviously the main one. The main one is designer babies and like creating the like ultimate best race of humans.
I mean, yeah, that one. I don't know if that one is a conspiracy theory, like necessarily, or very heavily fed by the fact that actually some of those experiments were being done. And I don't know who told me one that I was like, sometimes you think about those experiments that look like science fiction, that you will be like, we will engineer the babies to have blue eyes, like, and you're like, oh, maybe somebody somewhere is doing it, like without we know the problems of that, and you will never do it. But they're like, the world is very big, and people have different ways of seeing things and seeing progress, and other people have very little ethical considerations, so or very little morality. So they might be like, okay, whatever, let's try to. So I think those things were probably fed with this situation of the CRISPR babies, and I am afraid that I cannot say yes or no. I always say, like, it's illegal. It's not something that people will do in the open. What is being done behind closed doors? Nobody knows. But and that's, I mean, I think that nobody, like, I don't know. We will see when it happens, if it happens.
Yeah.
But that's, I don't think, I mean, seeing the president that we have, I wouldn't qualify that as a crazy conspiracy theory anymore, because it was like, I mean, the scientists went to jail, but that it put the president out there, which, yeah.
Okay, great. To wrap it up here, 2 more questions. How do you stay motivated and inspired in your work, particularly during periods when progress is slow or difficult?
I like to think, and I say that to my students, that you need to enjoy the science that you do every day. If you focus on, I'm gonna get this paper out in this journal, I'm gonna get this, this grant, I'm gonna go and be postdoc in this lab, and those are goals that, it's good to keep them in mind, but that might not only depend on you, and that they are very long term. And science, we cannot change nature. We can only go around and discover it. So you need to enjoy all that discovery process. You sit in tissue culture in yourself for however many hours, and you need to stop thinking like, yes, but this will give me the result of my Nature paper. I'm thinking like, all right, well, this is my hypothesis. I think this is the best way to test it. I'm very excited to see what it comes out of that experiment, and that with the small results and like the thinking process and the creativity and building a hypothesis, being right, being wrong, like pursuing things that you are happy and that you enjoy all that process. And then, if that ends up publishing in next way, like that's collateral, that you enjoy the process of doing science, the process of discovering, on coming up with ideas. I think that's, I was like, I'm usually not lacking motivation because I keep on going like that. And because if not, like science, and the more you go up, I think the worse. You have like everybody has like grant rejections, everybody has paper rejections, everybody has like fellowship rejections. You apply for PhD positions in like 5 universities, they say 5 no's, and you're like, okay. And that does not diminish your value, and that does not diminish how good of a scientist you are. It's just like, sometimes this is a competitive field, and it's not designed many times it's not designed for everybody to get the grants. It's certainly not designed for everybody to get the grants or to get the papers. But that doesn't mean that we are not enjoying what we do in our everyday basis.
Yeah, great. And lastly, what advice would you give to young scientists who are interested in pursuing a career in genome editing?
They should be prepared to like feel like they are behind the field all the time, like every day, more things. And I'm like, it's such a fast evolving and fast paced field that you always, my feeling, I'm like, another paper, I haven't even read the one that came out like yesterday.
Yeah.
It's there's so much research on that. It's like, and very fast paced. I always also feel like, even as a researcher, you are always going to feel a bit behind. You start your PhD with this model, with this Cas model, with this editor, and then during the duration of your 4 years of PhD, there are going to be like 20 different variant versions, or like 50 or 100 different variant versions. And you're like, why am I using the one that I had like 4 years ago? But, you need to find a compromise, find the best one, and find the one that gives you the answers, because it's like you're always going to be behind the people that develop technology if you are working on applying technology. And you have to be, good with that, good with keeping up what is coming out there, and good with like making decisions and compromises on like, okay, I think I'm going to go ahead with this, instead of like trying to constantly see how can I make this better and better and better and better, because, there are people working on that research. And
so how do you like try to stay productive with like juggling all of these things, from like I don't know, mentoring your students and like doing experiments, meetings, like outreach and all of that?
That's well, I try to organize myself. Yeah, I am quite thorough on organization, because it's very easy to to lose track, and I think you have to have very clear what is important and what is less important, and especially I think at the stage of the new investigators, be like, protect your time and also take care of yourself, because I would say like science is something that never stops. So if you want to work more, you can work more. So when I have like already a long day, 12 hours, and I leave the lab and I'm like, I haven't done anything. I have these 3 classes, whatever. Like, I was like, what did I do today? And you did a lot, but the pile of work is never ending. It's still there. I could write one more grant. I could do more. There are always things, but you need to learn when to say yes and when to say no. And, and also for students, you mentor them, and then you also need to let them fly and make their own decisions and make their own mistakes. You cannot be micromanaging them all the time. So, it's I don't know. I'm learning. I'm like I think we never stop learning, and I have just started. So it's a learning process for me as well. So.
Yeah, that's great. Thank you so much. I think it was great. Any last words?
No, thank you. Thanks to you, Mike.
My pleasure.
It's my pleasure to be here. It's been a great interview, and I look forward to seeing it, and also to seeing many more of your future guests, and all the success for you in your podcast and in your master's and future career.
Yeah, thank you so much.